can go ahead and start, and people can join in as and when they are coming in. So once again, welcome everyone to Speedy Science 2021, organized by Phi Tau Sigma, Honor Society of Food Science and Technology, and sponsored by AgroPure. And before we start the competition, we would like to run through a couple of outline of our agenda. So we'll be starting by introducing Phi Tau Sigma, who we are, what we do, and then move on to the introduction to Speedy Science 2021, and also a thank you note to the sponsors. And then we'll be moving on to the finalist presentation, and you'll be hearing further details about trivia too. So we have an audience engagement trivia in Kahoot. So everybody who is on the participant, please be ready with your phones. And you'll be hearing more details on that throughout the competition for the trivia as well. And finally, we'll move ahead with the winner announcements. So Phi Tau Sigma, so who we are, the Phi Tau Sigma UMN chapter. We are a chapter which began in 2013, and it's run by students in the food science department with the guidance from Dr. Mary Schmill. And it's one of the chapters of the national or international Phi Tau Sigma. And Phi Tau Sigma recognizes the scholarly accomplishments and awards of food science and technology students and professionals who excel in their field of study. The reason we are unique is because we stand as a collaborating chapter, allowing students and professionals from UMN, NDSU, and SDSU to be part of the National Phi Tau Sigma. And members serve in other leadership committees, including IFT, ADSA, AACCI, ASABE, and COFFEE as well. And we are part of several successful meetings and competitions. And in addition, there are also parts where student scholarships and competitions and awards are also being given to members, student members, as well as professional members. With that being said, we would also like to extend a warm welcome on behalf of all our members, board and professional members, to be part of this Phi Tau Sigma UFM chapter. And being a member will provide you lots of benefits, eligibility for awards, scholarship, leadership development opportunities, and you will also have a lot of networking and mentorship opportunities and competitions like Speedy Science as well. And you'll also get you'll also have a chance to get featured in the national newsletter where thousands of people read the newsletter. And finally, we'll also be giving some honor cards and prints for your graduation, as you can see in the pictures we have shown. So feel free to email me or contact us if you have any questions on how to join Phi Tau Sigma, and we'll help you through the process. Be the best and the brightest. With that, I would like to present to you the officer team of 2021, and without whom this competition is the first time we are doing it, and we hope we did a good job, and we love to hear feedback from you guys as possible. And I'm Sonali Raghunath, and I'm a PhD student, and I'm the current president for our chapter. Moving on, we have Yara Benavides-Paz, and she's a PhD candidate and our current co-chair and also the past president of our chapter. And next we have Vaidehi Narkar, and she's an MS candidate, and she's our vice president. And then we have Akua, who's a PhD candidate, and she's serving as a treasurer. And finally, we have Radhika Bharati, who's a PhD student. She's serving as a secretary and also the co-chair for the Speedy Science 2021. And she's given tremendous effort in putting this together, and we hope to see all come true today. And without any further delay, let's go on to Speedy Science 2021. And we would also like to thank AgroPure, especially for coming forward as a gold sponsor for this very new event initiative. And we are very happy and grateful to have AgroPure as our sponsor for our Speedy Science event this year. And this event is definitely going to be a starting point to many amazing future scientists, food scientists, and thank you for helping us to make it happen. And we'd also like to thank our bronze sponsor, Danielle and Erica Kennedy, for coming forward as an individual sponsor for this event. And once again, thank you sponsors for making this happen. Next, we also have three amazing judges for the competition. We have Dr. Mary Schmill from University of Minnesota, and Dr. Muthukumarapan from South Dakota State University, and Dr. Yu Jong Lee from University of Wisconsin Stout. And I really like to thank them for agreeing to serve as judges for this competition and taking their time. With that, I would like Radhika to take over, and she'll be moderating the rest of the session. And good luck for all the finalists.
Thank you, Sonali. Hey, everyone. My name is Radhika, and like Sonali mentioned, I will be co-hosting the event with her today. Before we begin, a couple of basic housekeeping rules. I'm sure we're all very familiar with Zoom, but this is protocol. So for all the audience members here, make sure that your microphone is muted, especially when the finalists are presenting, so that we don't have any noise or interference. However, we have a live Q&A event planned after each finalist's presentation, and at this point, you can either choose to unmute yourselves or put your questions in the chat. We will be monitoring the chat and watching out for any questions there. And finally, we would love for all the audience members to show your support and encouragement for your peers presenting today. Zoom has these really cute reaction icons. You should see some popping up on my face right now. Feel free to use these or put in your wishes in the chat. And it's always good to encourage your peers and show them your support. All the finalists have worked very hard for this competition. Moving ahead, to give you a general outline for the event, we have a pre-decided presenter order that we've shared with the judges and the finalists, and we will be following that. After each finalist presents their talk, we have a short Q&A session. We have to be mindful of time, so we'll first open the Q&A to the judges and then maybe take 1 or 2 questions from the audience. However, if audience members have questions, they're free to contact the participants, have their email IDs, and we'll get you in touch with them for later references. Then while the judges are scoring the participants in real time, we have some fun trivia planned for the audience members. So thank you for joining us. And we're using Kahoot.it. For those who don't know much about Kahoot, we will walk you through the Kahoot website and how to log in and all of that. But for now, keep your phones or your laptop browser handy, and we have some fun time planned for you. With that, let me call on stage our first speaker for today, Lauren Avalos. She's a master's student studying food science at the University of Minnesota. Her talk today is titled Leaving a Bitter Taste, Are Panelists Waiting Long Enough During Taste Testing? Lauren, I welcome you on stage, and over to you.
Thank you. Get my, okay, you should see my screen now.
Yes, and we're ready whenever you're good to start.
Great. Okay, so let's set the scene. We're totally post-pandemic, and you and your friend are at your favorite Mexican restaurant. The waiter stops by to take your order and drops by some chips and salsa. You know you've still got dinner on the way, but you still find yourself shoveling that spicy salsa into your mouth. Did you happen to notice that as you dug deeper and deeper into that spicy salsa, it just kept getting spicier and spicier? The heat just kept on building until your mouth was totally on fire. Well, there's actually a scientific reason behind this fire in your mouth, and it's the focus of my research. But I didn't study chips and salsa. I studied how time between bites changes the bitterness intensity in Brussels sprouts. And I know that's not quite the same thing as the fire in your mouth, but bear with me, it is just as important. So if we keep with this chips and salsa example, as you were shoveling the chips and spicy salsa into your mouth with barely a breath between each bite, your friend across the table from you was pacing themselves. Between each chip, they took a breath, a large sip of water. Maybe they switched between mild and spicy salsa. As they ate their chips, they were clearing their mouth between each bite, and the fire of spice never built up. This is shown on the figure in the upper right-hand corner of my slide, where you are the dashed red line that increases intensity with every bite, and your friend was able to maintain a relatively flat line with the blue line and did not build up spice. This is the same idea as wait time and sensory testing. If a panelist allows sufficient time for the food item to clear from their mouth, the taste will not accumulate into a fire, or in the case of my Brussels sprouts, a very bitter coating in their mouth. For my study, I looked at how long panelists waited between Brussels sprout samples and what happened to the bitterness ratings over the course of the study. Panelists were told to wait as long as needed to clear their mouth of bitterness or Brussels sprout flavor between bites. I found that panelists did not wait longer after tasting more bitter samples, but that there was still no buildup of bitterness. The first part of that finding, that panelists did not wait longer after tasting more bitter samples, is shown on the bottom left-hand corner. If panelists had waited longer after more bitter samples, we would expect to see a positive slope to the data, demonstrated by that purple dashed line. But instead, we see a horizontal line, that bold black line, that's nearly parallel to the x-axis, indicating no relationship between bitterness and time waited. The second finding, that even though panelists did not wait longer, they still waited long enough to avoid buildup, is shown in the bottom right-hand corner. There's no difference in average bitterness from the start to the end of the study session, and that slope of the line is not significantly different from zero. If it had been, we would have expected a positive slope. These findings were strange. I had guessed that, like, you and your friend eating salsa, people would wait longer after more bitter samples. My research shows that different food items and different types of taste may need different approaches to the time waited between samples. Thank you.
Thanks, Lauren. That was a fantastic talk. You may now stop sharing your screen and we can take questions for Lauren now, beginning with the judges.
Hi, Lauren. Can you hear me? This is Mary.
Yes.
Oh, great. Thanks for that great presentation. Really enjoyed it. Now, you talked a lot about using like salsa as an example, but you actually tested Brussels sprouts?
Mm-hmm. Yeah, okay. So I...
Like, why is that? Yeah.
I found that it was easier to get people to understand this concept of buildup when they could think of something that they can probably have experienced. I mean, most of us probably have dug into something spicy and noticed that buildup over time. It's just not quite the same as, like, buildup for bitterness isn't as extreme as spice. So the thing that I was measuring was much more subtle than like a bowl of spicy salsa would have been.
And just a quick follow-up question, like what do you plan on testing next? If you like chose Brussels sprouts, like which food would you want to just test next?
If I was to do something like this again, I would probably do a model system. So using like solutions of caffeine where I can really control the bitterness levels. That was one of the limitations of this study, is that I wasn't actually able to go back and see if panelists were accurately discriminating samples because I didn't necessarily know what the bitterness of the Brussels sprouts was before the study. So being able to do this with a model system of caffeine solutions that go from very bitter to not at all bitter would have allowed me to do a little bit more analysis to check panel performance as well as this build-up concept.
Okay, thank you.
Any more questions for Lauren from the audience or judges? Okay, if not, I have one for you, Lauren. You studied the buildup of bitter flavors, but you gave an example for salsa, which is a spicy. Mm-hmm. So do you think buildup is the same for these different types of flavors that you see in bitter versus spicy versus sweet?
So no, it's similar and it gets at the same point, but buildup of spices, a spice is actually a pain receptor. So it's a little different in terms of how that affects your actual, like, the chemistry in your mouth, whereas bitterness is a protein receptor in your mouth generally. And so they're slightly different. Again, like this, my use of the chips and spicy salsa was really to try to make this a little bit more relatable. I don't think that many people are eating a bowl of Brussels sprouts and noticing how bitter they get over the course of eating it. So
thanks. Yeah, that makes sense. And salsa is definitely making it more relatable than Brussels sprouts. Thank you, Lauren. So while we let the judges grade, we now have a trivia planned for the audience. So all audience members, please go to www.kahoot.it, either on your phone or laptop browser. And in a few seconds, we will be sharing a game pin with you that you would need to enter in your Kahoot website and also put in a nickname. And just as a reminder for those who are new to Kahoot, you need to come back to the Zoom screen to see the questions, but you need to use your phone or your laptop to select the options. So if you're looking for where the questions are, please come back to this browser and you'll see them right here. With that, let me quickly share my screen to bring up my Kahoot. Okay, you should hopefully see my screen. So audience members, this is your game pin for today. And I look forward to seeing you right here. Jana, welcome. Give it a couple of more seconds before we start. Okay, so let's begin with our first trivia question for today. A lipid chemistry question. Okay. Let's see who is on the top. I see that the other participants have, are you able to access Kahoot? If you have any issues, please let us know in the chat and we can help you troubleshoot. Okay, going to our second trivia question. Flavor chemistry. What do you think? Spicy aromas. Ooh, Cheese Masters on the top. Okay, so let's go back to our second speaker for today. But for all the audience members, please do not log out of Kahoot. Think of this trivia like a marathon. So we'll be playing questions between every speaker. And let's see who's our trivia winner for today at the very end. So going back to our second speaker for today, Sonali, you should be able to share your screen.
Are you able to see my screen?
Yes, it's not in the presentation mode. Now it is. Thank you so much. So calling us a second finalist for today, Kavya Ratnakumar. She's a doctoral student studying dairy science at South Dakota State University. Her talk for today is titled Smart Solvents in the Application of Milk Phospholipids. Welcome, Kavya, and the stage is all yours. You may now share your screen.
Can you see my screen now?
Yes, Kavya, we do. So whenever you're good to start.
Okay. Very good. Just give me a minute. I couldn't. Since I'm using like two screens, I couldn't do my
Take your
video.
time to figure it out.
Okay. Yeah. A very good evening to everyone present here, and I'm really glad to be part of this 3 minutes thesis and excited to present my work here. As we are in the new normal, the consumer trends on eating more healthy and nutritious food that enhances the immunity has come along the way these days. Therefore, the dairy products and the byproducts serves this purpose naturally. Who doesn't enjoy eating ice creams? It is even nicer and enjoyable when the ice creams is enriched with healthy ingredients that can benefit people from all age groups, especially during this time. The functional ingredient that we have enriched with the ice cream are the phospholipids. These phospholipids are naturally present in the milk fat, and during processing, they go into the dairy byproduct stream. For instance, buttermilk, beta serum, procream, all these low-value byproducts contain an enormous amount of phospholipids. Consuming these phospholipids helped in reducing cardiovascular disease, improved immunological functions, cholesterol absorption. In recent times, it is used in the infant formula for the growth and brain development of the infants. However, the challenge lies in isolating these phospholipids. The techniques which are previously in use involve a multi-step procedure. They use a lot of chlorinated and flammable solvents that are injurious to health, and they also use a distillation process that causes environmental hazards as well as they are economically unviable. So, in a smarter and a sustainable way, we came up is the use of switchable solvents, which are amines. The specialty of these solvents, they can switch on and off from water immiscible form to water miscible form by simply bubbling the carbon dioxide. When you remove the carbon dioxide, it goes back to its original form, and the magic lies here. You can use the solvent for several extraction process. It is reusable. From this technique, we were able to obtain 99% of phospholipids, a remarkably high yield compared to any other traditional methods and the techniques which are in existence. And further, these phospholipids were enriched into the ice creams. In an economic point of view, this process is highly beneficial, starting from a low-value byproduct and converting to a high-value end product of phospholipid-enriched ice cream. This work can, throughout maintaining a sustainable process system as a whole, this work can help the dairy farmers in generating more income and also creates more business opportunities to industries by increasing the utilization of the dairy byproducts more effectively. Thank you.
Thank you, Kavya. That was a fantastic talk. You may now stop sharing your screen. And judges, we can take questions from Kavya. So open to you.
Kavya, this is Muthu. Excellent job with your presentation.
Thank you.
So my question is, when you say your solvent can be reused,
yes.
How much solvent can be reused?
So the whole amount of solvent, it can if you are taking a 20 ml of solvent for the extraction process, and then we remove the layer of phospholipids, it's like when it is switched, it's switched, so it goes to like an ammonium bicarbonate salt, which is dissolvable. That makes the phospholipids or the lipids layer on the top. So at that point of time, we didn't exactly record the recovery, but then of how much amount, like maximum like 10 to 15 ml of solvent, some 15 ml of solvents we can recover, but we have not exactly known that how much recovered it can be, how much the use of recovery in further extraction.
Okay. What type of solvent is it? Is it green solvents? Is it industrially used?
So it is a dimethyl cyclohexyl amine, which is approved by an FDA as an indirect food additive. And yes, it is a green solvent because evaluating the toxicity and all those things compared to other organic solvents such as chloroform, methanol, it was synthesized in the laboratory, and yeah, they are green solvents.
Thank you.
Kavya, we have one question in the chat,
Yes.
but I heard someone talk, so please go ahead. We can hold off.
Yeah, this is Mary. Hi, Kaya.
Hi.
I'm trying to remember my biochemistry. So phospholipid is a triglyceride, is part of the composition, and phosphate and choline sometimes, or maybe serine. But within the composition, what is it about it? Is it the structure, or is there some unique fatty acids, or what is it in the phospholipid that might help it have special health benefits for the human body?
So this whole milk fat globular membrane, as a whole, it contains the proteins are complexed with this phospholipids. So what we try to do is like destabilize the proteins and obtain these phospholipids. And these phospholipids have individual phospholipids, which is based on the double bond and the saturated chains, which can lead to like different individuals such as sphingomyelin, phosphatidyl serine, phosphatidyl choline, phosphatidyl ethanolamine, and sphingomyelin. So that the exact one, this can be quantified using HPLC, which we also did that. And the most bioactive phospholipid are the sphingomyelin, which is prominent in the milk fats, and that can be used as it's used in the infant formulas in the recent times for the grown, and it's more on the nervous development, and also it acts as an anti-inflammatory and all those other health benefits with those.
Thank you.
Kavya, we have one more question in the chat that I can read out right now. Is this solvent method currently used for any industrial products?
No, it has not yet used for the industrial products, but then it has been used in the algae research, soybean oil processing. This was in a way a sustainable way to reduce the distillation process in the industry. So it has still, it is not existence in the industry, but it has been used like in the other research.
Thank you so much. Yes, that makes sense. So while we leave the judges to grade, let's go back and continue our trivia. So all audience members who have joined in late, this is the game pin right at the bottom of my screen, and it's also in the chat. One of my team members will post it in the chat. Please go to www.kahoot.8 and you can join us right here. And as an incentive, we have exciting prizes and gift cards for trivia winners today. So let the show begin. The third question for the trivia: when was Phi Tau Sigma established? Correct answers. Nice. Cheese Master still on top. How many of you remember membrane separation technology here? Four correct, very nice. Oh, I see some new names coming up. Good job, guys. Okay, I am going to stop sharing my screen, and assuming that the judges are done with grading, we can then move on to our third finalist.
Just going to give a couple of minutes for the judges.
Okay.
Okay, I think we're good. We can go move on to the next participant. All right, are you guys able to see my screen?
Yes, and I think we need to go on. Yes, correct. Thanks, Sonali. So our third finalist for today is Rebecca Schulz. She's a master's student studying food science and nutrition at the University of Minnesota. Her talk today is titled The Carbohydrate Saga, using a systems lens to define carbohydrates quality, including additional metrics to maximize human health and environmental sustainability. Welcome, Rebecca, and you may share your screen now. Yeah, we can see your screen. It's good.
Are you able to see?
Yes.
Okay.
Whenever you're good to start.
Okay, and we'll begin. So my name is Rebecca Schulz, and you can see my title in the left-hand corner of the screen and further broken down into my diagram. So to begin, I'll draw your attention to the blue 1 at the top of the circle. We'll just frame this presentation up with why we are talking about carbohydrates in the first place. Historically and to this day, carbohydrates have made up both the majority of agricultural commodity production and comprise 55% of our dietary intake in the US, according to N. Haynes. While there's been a vacillating degree of acceptance for carbohydrates in the US, carbohydrates are here to stay, and even more so, as we realize that in order to meet specific climate goals, we will need to increase plant consumption. Additionally, we know that what we eat is intrinsically linked to our health. So bottom line, carbohydrates matter. And if they matter, wouldn't the quality of these carbohydrates be of utmost importance? But before we get into defining what a quality carbohydrate is, let's talk about how we decide what a quality carbohydrate is first. We'll move to the right to the green 2, where you can see the contrasting siloed reductionist approach versus a holistic system approach. We're beginning to discover that things do not operate in isolation, but rather systems. And thus, as we position our fields and departments in a similar manner, meeting multidimensional problems with multidimensional solutions will maximize results and operate as the world actually exists. Okay, so now taking this systems lens with us, we'll move to the red 3 to define carbohydrate quality using this systems lens. Historically, the 4 metrics used for carbohydrate quality have been whole grain content, fiber, added sugar, and glycemic load/index. These metrics should continue to be included in a future definition, given epidemiological studies linked to health outcomes. However, we argue that they should be incorporated using a standardized algorithm, as up until now, these metrics have been applied arbitrarily, yielding a non-holistic approach. From here, we'll move to the yellow 4 at the left hand of the circle, bringing these 4 metrics with us. But in lieu of systems thinking, we also need to think of broader concepts, such as sustainability, to include in a holistic definition of carbohydrate quality. How will our definition impact reference diets, agricultural production, and therefore our ability to reach goals such as the Paris Agreement? Thus, a sustainability metric is needed. Additionally, how the food is prepared matters just as much as what it contains. So including degree of processing, referring to ultra processing, and provided nutrients of concern as measures to consider. This brings us then to the center, which is at the heart of why we're doing all of this in the first place. As we've mentioned, as we create a comprehensive, holistic, systems-based lens to define carbohydrate quality, we will not only be impacting and improving human health, but also environmental sustainability, ensuring that those who come after us will have the resources they need to flourish and survive. Zooming out, you can see by this continuous circle that these things all operate in a relationship with each other, and we should too. Here's to future systems, collaboration for maximum results for human health and environmental sustainability.
Thanks, Rebecca. That was a great talk. You may now stop sharing your screen and we can open up for a live Q&A session. So beginning with the judges. Questions for Rebecca?
Hello, Rebecca. This is Julie, and it's a great presentation. So this concept is just like that you just combine with the health, the quality, and then the environmental quality together, and then the judging of the carbohydrate quality. So the depending on the grain or the carbohydrate source, all is just the same trend. I mean that between the health, the quality, and then the environmental quality is the same trend, or totally different. How about that? Is the individual grain is that similar or different?
Yeah, and let me know if I'm not answering your question, but we're seeing a similar trend. So both increased plant consumption is going to benefit human health based on epidemiological studies, looking at those parameters that have been defined thus far, as well as we're seeing, and you can use a variety of goals as the endpoint, but agricultural production produces a ton of greenhouse gases that are going to accumulate and contribute to climate change. And so what are ways we can kind of mitigate that? And we're seeing that animal products are kind of a large part of why that gas production is there. And so if we can minimize that and move in these plants kind of as a larger part of a reference diet, then we can both improve human health and also improve kind of these climate parameters. So to answer your question, we're fortunate in that they trend together. It's just a matter of, can we come up with a straightforward way to define kind of the indices to include, so that we're recommending things that are going to allow for that trending together. Yeah, does that answer your question?
Yeah, and then one of the following up question is, would you just explain one of the example how you can measuring the environmental or sustainability of those grain?
Yeah, definitely. So different folks have talked about using, you could use a variety of parameters under the umbrella of sustainability, if you will. So carbon footprint would be one, for example, or life cycle assessment. So kind of from the minute it's in the ground all the way till it's on the consumer's plate, what are the miles involved in the travel and the gas used in the tractor and the inputs into the field, et cetera. And so different people have talked about, we pull up an NDSR, kind of like the nutrient profile of a food. What if alongside that was an indicator of its carbon footprint, which is pretty onerous to think about entering all that data, calculating all of that data. But if we need to be strategic in how we're meeting these goals, and if there's not a lot of margin, the effort is maybe worth it, because we'd be able to have more of a straightforward and defined approach to how to reach that.
Thank you.
Hi, Rebecca. Mary,
Hello.
just a quick follow-up question on that. Like I used to see that a few products would have a carbon footprint, like we have nutritional labeling. I mean, do you envision carbon footprint information like nutritional labeling on the label in the future?
Yeah, that is something that's definitely been talked about. I know in my agribusiness class, we've talked about like transparent labeling. So similar to how there's like a fair trade label or non-GMO, whatever you want to put in there, perhaps there could be a carbon footprint label, which you'd have to, co-teach along with that, so the population can understand, what does that even mean? What's a better versus worse? What's something, like, how can I average that for my home? Like, I think there'd have to be a lot of education around how a consumer is to kind of interact with that label, but it could be beneficial. I mean, we see increased labeling around other measures to increase kind of those endpoint goals. So perhaps if an endpoint goal is improved climate outcomes for these products, that would be a valid way to kind of reach that endpoint.
Thank you.
Thanks, Rebecca, for answering all the questions from the judges. If audience members have any questions, as a reminder, you can always put them in the chat and the finalists can reply back to your questions back in the chat. So while we let the judges grade, let's go back to playing trivia again. Question number 5. What do you think? What does the brain need? Wow, most of you got it right. Good job, Ahmad. You seem to be on fire. Question number 6. When was the microwave introduced? Some good guesses there. Ooh, we have a new lead in now. Azul, good job. What do you think was eaten on Moon? I didn't know this either. Ahmad is on fire. Good job. Ooh, a streak of 5 correct answers. Wow. Okay, so let's go back to our third finalist, assuming that the judges are done with grading. Giving it a couple of seconds. Yeah, I can see your screen now, Sonali.
Fourth finalist.
Fourth finalist, sorry. So our fourth speaker for today is Mohammad Al-Farouk. He's a doctoral student studying dairy science at South Dakota State University. His talk today is titled Encapsulating Bioactive Compounds for Improving Human Health. Welcome, Mohammad, and the stage is all yours. You may now share your screen.
Do you see my screen now?
Yes, we do, Mohammad. So whenever you're ready to start.
Okay, thank you for the introduction. Hello, everybody. As you are aware, there are a lot of biological bioactive compounds available in nature. We could improve our health by consuming them on a regular basis, but there are some roadblocks. To explain, you all like grapes, right? Did you ever have a chance to bite the grape seeds? I did. We could look younger by consuming grape seeds. Sadly, the current generation is not getting a chance to experience the better taste of grape seeds. Grape seeds contain one active molecule called resveratrol that has the ability to slow the aging process and reduce the cancer risk and inflammation. Similarly, turmeric buds are being used as a spice our colorant foods. Its active ingredient, curcumin, is healthy. Its consumption with hot milk helps to prevent cold issues, especially during the winter season. However, these compounds incorporation in foods is not easy. They do not dissolve in water, and they are heat, light, moisture, and oxygen sensitive. Furthermore, they become inactive during the acidic condition that we normally encounter in our stomachs. In this regard, these compounds need to be protected by using suitable material. My research focuses on carrageenan, which is an FDA-approved and generally recognized as a safe carbohydrate material. Our carrageenan is found to enhance the solubility of resveratrol and curcumin and protect them from the heat and oxygen. After encapsulating resveratrol and curcumin in the carrageenan matrix, we are developing healthy foods as breads that incorporated with these complexes. In order to gain full health benefits of these bioactive compounds, we should have a clear-cut picture about their release in the stomach, and the stomach conditions and GI tract. Toward this end, we created simulated conditions and we recorded the release profiles. The results show that the majority of release is taking place in the intestinal condition than the gastric stage. In the final analysis, the used wool material carrageenan is could protect resveratrol and curcumin from the harsh conditions in the stomach and safely pass them to the intestine gaining maximum health benefits. With that, I would conclude my research, and thanks for your attention.
Thank you so much, Mohammad. That was a really great talk. You may now stop sharing your screen, and we can open up your live Q&A session beginning with the judges.
Okay, thank you, Mohammed. I'm going to run out and get some grapes and I'm going to get younger and healthier overnight.
Yes.
No, I really enjoyed it so much. Are the compounds that you talked about, are they sensitive to oxidation, or what is the reaction that breaks them down?
So they are first of all, they are phenolic compounds, or they have like the functional groups. They will react with the oxygen and will change the activity or the bioavailability of these bioactive compounds.
Okay.
Thanks, Mohammed. Any more questions for Mohammed from the judges or the audience?
So would you explain just provide the example what kind of the compound or material you can use with the encapsulation material?
So in my case, I am using carrageenans. So I'm using carrageenan in my case. And also I add some other compounds like a little bit of sodium chloride. And also I am using different type of cyclodextrins to enhance the encapsulation and release profiles.
So to make the encapsulation, you also using some of the instrument or equipment, kind of the spray dry or?
Oh, I'm preparing gels. Then I make fibers. Those fibers are used to encapsulate the bioactive compounds.
All right.
So,
yeah.
Thanks, Mohammed. Any more questions for Mohammed from the audience?
I have a question for Mohammed. So what other food products do you think will add these bioactive components? I do know that you mentioned bread. What other food products do you think that this can be added into? And yeah, that's my question.
Right now, I am planning to add them to dairy products and see what will happen to those encapsulated bioactive component and the product itself.
Okay. And how much of a bioactive component would be needed to be added? Like, do you just need small amounts, or do you need more of it to be effective in a food product, like a bioavailability?
According to the release profiles I have collected, we don't need a lot of the bioactive component. Small amount of the bioactive component is enough to get like enough amount of bioactive component when you consume the product. So, but still, I don't know how much I should add to be able to say it is like good enough to get this amount of this bioactive component or not, because we're still using like just the in lab experiment. We did not try it with animals or try it with human beings. So it's still under like the process.
All right, thank you.
Thank you, Ahmad, for answering all the questions. And now, while we let the judges score, we can again go back to trivia. I see a couple of new members joining in. So for everybody who's joined in, you can always join the Kahoot. The game pin is in here. And we will wait a few seconds to see if we have any new members joining in on the Kahoot before we go to our next question. If any of you have issues with Kahoot, let us know in the chat. So let's move on to the next question then. Yeah, our backgrounds gave the answer away here. So you should have got this, I'm sure. Ooh, Azul is on the lead. Ooh, and Jacob has a streak of 5 answers. Good job. To all the sensory gurus in the house, I'm sure this is a cakewalk for you. Oh, I wish we could do sensory during, hopefully soon after COVID. Lauren, of course, this was a sensory question. Okay, so let's go back to our 5th speaker for today, assuming all the judges have turned in their responses. Our 5th speaker for today is Sarah Kleber. She's a doctoral candidate studying food science at the University of Minnesota. Her talk for today is Chewing Down on Cheese. Welcome, Sarah, and the stage is all yours. You may share your screen now.
All right. All right. Can you see it yet?
Yeah, it's not in the presentation mode yet, though. We can see it now, so whenever you're good to start.
So, hello, once again, my name is Sarah, and for the next 3 minutes, I'm going to talk to you about nothing but cheese, specifically enzyme-modified cheddar cheeses, which are cheddar-based flavorings made by adding mixtures of enzymes to cheddar cheese and using special processes to speed up the cheese aging. And what's great about these cheeses is that they can be produced more quickly than normal cheese, and they can be added in very small amounts to a variety of foods to create what I lovingly refer to as a cheesy flavor explosion. So these cheeses definitely have their advantages, but a major issue is that their sensory and chemical properties have not been extensively researched. And this can result in product inconsistencies and increase the amount of time and money spent during product development. But fortunately, the findings from my research, which is focused on characterizing the taste, flavors, and aromas of 9 enzyme-modified cheddar cheeses and identifying relationships between the sensory and chemical properties, will allow manufacturers to produce specific flavors and aromas in enzyme-modified cheeses and to provide their customers with eternal happiness. So now you're probably thinking, that's great, but how did you do this and what did you find? And since you asked, I'll tell you. Well, I began by recruiting 11 participants to attend a series of 1-hour sessions across a period of several months. During these sessions, I had them taste and smell my cheeses and provide terms they would use to describe them, such as nutty, fruity, and yes, vomit. And once we had compiled our list of the taste, flavors, and aroma terms, the participants completed 2 days of testing in which they were given each cheese in duplicate and rated the intensities of the different aromas, flavors, and tastes they had detected over the past months. And using these intensity measurements, along with chemical information provided by the cheese's manufacturers, and completing what I like to call stats magic, we found that of the 44 different aromas, tastes, and flavors the participants had identified, 14 aromas, 2 tastes, 6 flavors, and astringency differed among the cheeses. Furthermore, by looking at these differences, we found that the harsh, more pungent flavors and aromas, such as blue cheese and horseradish, tended to be more intense in the cheeses with more free fatty acids and more amino acids. But the mild and less aged flavors and aromas, such as butter and dairy, tended to be more intense in those cheeses with a higher pH and greater % fat and protein. Therefore, it seems that if manufacturers want to produce a cheese with the harsh, more pungent flavors, they need to generate a lot of free fatty acids and free amino acids. But if they want a more mild, less aged cheese flavor and aroma, and they really want that to shine through, they need to maintain a high pH and a high % protein and fat. And that's my speech. Thank you.
Thank you, Sarah. That was a fantastic talk. If you will, stop sharing your screen. We can now take questions for Sarah, beginning from the judges.
Hi, Sarah. Thanks for your presentation. It's an excellent presentation. Can you hear me?
Yes.
Yeah. You talked about the flavor enhancement and also the you can use only less amount of cheese with the specialized cheese.
Yes.
How about the melting characteristics? Is this cheese smells better than other cheeses, or what is the functional characteristics of cheese?
So we did not look at the functional characteristics of the cheeses in this, but they're very similar to like a paste that you would add to different products. So it's almost like if you took the powdered cheese from like boxed mac and cheese and mixed it with water or something like that, that's similar to the consistency you would get. But we didn't really look at the texture of these cheeses. I guess, is that what you're asking?
Oh, that's okay. Thank you.
Okay.
Hey, Sarah, Mary. So great presentation. Yeah. Makes me want to go out and get some Cheetos or something. Because I really like that. You talked about that cheese explosion.
Yes.
Do some manufacturers who
you said seed, right? Yeah.
Do some manufacturers who are want that cheese explosion, are they, because you talked about it in your talk, are they like adding some free amino acids or maybe some protein hydrolysates or free fatty acids to get that flavor?
Well, currently they're not doing that because they don't really understand what's happening when you add the enzymes to the cheeses. So they try to control how many free fatty acids and free amino acids are present in the cheeses with the enzymes they choose and the processing parameters. But I don't believe they know specifically what kinds and what the balance between the free amino acids and free fatty acids to achieve those specific flavors, which I think makes it difficult just to add those components.
So you're really on the cutting edge of trying to understand like all those flavors that are development, and then you got to find out which types of free fatty acids or how to produce them in the cheese.
And mainly focusing on the overall composition of the cheese as opposed to just, can we link this specific compound to this? It's more looking at if we have a lot of free fatty acids and a lot of free amino acids, that is how we are able to produce those harsher flavors, as opposed to looking at specifically, we need a lot of just this one free fatty acid to get that flavor. So it's a lot of what we're doing is looking at, it's a balancing act.
Yeah, appreciate that. Thanks.
Sarah, so that you said that you they use 11 participant for this sensory, right? So before you starting this project, did you do some special training or screening method for the participants?
Yes, so we do screen our participants to make sure that they can taste bitter compounds, which we call them probe tasters. So we make sure that they're able to do that. We also give them tests to see their abilities to describe texture, flavor. Can they discriminate between different intensities of flavors and aromas? And that's how we get our pool of panelists. And that's how I selected the 11 participants I would have.
Thank you.
Sarah, I have a question. So enzymes are going to be specific. So why is it difficult to know quantitatively what free fatty acids are being released in the cheeses?
Well, you can measure the quantities that are being released, but there's still a lot of unknowns because cheeses are so complex. A lot of factors affecting what the enzymes are actually acting on and what compounds they're going to act on. They're specific, but there's still so much unknown about how everything is in that food system to be able to understand the exact chemical pathways that are generating these flavor compounds.
Okay, thank you.
Thanks, Sarah, for answering all the questions. Do we have any more questions from the audience? We do not see any in the chat. So thank you, Sarah. We will go back to playing trivia and let the judges do the grading. Okay, a reminder to all the audience members again, it is not late yet. You can still join in on our trivia. Go to www.kahoot.it and use the game pin right here at the bottom of my screen. And we would love for you to join us. To all the vegetarians in the crowd, you should know this. Well, it is. Cheese Master back on top. Good job. 3 correct answers in a row. Impressive. Some food micro. What do you think? Lauren on the lead. Good job, Lauren. With that, let's go back to calling our 6th finalist for today. Our 6th speaker for today is Nadi Kaluwahandi. She's a master's student studying agriculture and biosystems engineering at South Dakota State University. Her talk for today is titled Effective Cold Plasma for Inactivation of E. coli and Quality Changes in Beef. Welcome, Nadi, and the stage is all yours. You can now pull up your slide.
Okay, give me 1 second. You can see it now.
Unfortunately, we cannot see your screen. You have to hit the green button that says share screen at the bottom. And then you have to begin. Yes, we see it now. So you can start from the top again.
Okay. Sorry about that. Good evening, everyone. First of all, do you know that the cold plasma technology is the future of quality control in food processing industry? So today I'm talking about the effect of cold plasma for inactivation of E. coli and quality changes in beef. So cold plasma is one of the non-thermal techniques which is well ionized non-thermal temperatures ranging from 30 Celsius to 60 Celsius with atmospheric pressure and requiring low energy. So my main objectives of this research are quantifying any nutritional changes of the beef due to cold plasma treatment and inactivation of food pathogens such as bacteria and degrading microorganisms concerned. For this research, I used the high voltage plasma system as a cold plasma generator. And then my methodology was I prepared bacteria culture and then I inoculated the bacteria culture on the beef surface and then treated the cold plasma with different treatment times and different discharge frequencies. Finally, after 24 hours, I counted the number of microbial colonies and calculated the log reduction relative to the control. So the research showed that when the treatment time increased from 1 minute to 5 minutes, and the discharge frequency increased from 2000 to 2500 hertz, the log reduction or the bacterial inactivation ratio was increased. So moreover, I have treated Salmonella sero and the Salmonella dublin, and I obtained 1.6 and 1.7 log reduction as well. So after the cold plasma treatment, there was not any significant difference due to treatment, like different in temperature, moisture content, and surface color. So in conclusion, I would like to say that cold plasma technology can be used as the food processing industry. So I would suggest that, like, as my next milestone, I would like to go with the cold plasma and another non-thermal technique for achieving a high frequency. I mean the high log reduction rate. Thank you.
Thanks, Maddie. That was a great talk. You may now stop sharing your screen and we can open it up to questions beginning with the judges. So judges, your questions for Maddie.
So my question is, you just only showed the one graph for the E. coli, but you mentioned that you also tested the Salmonella and other the microorganisms. So through this treatment, which one is the most the effective? You mean that the E. coli is the most effective?
No, the thing was I submitted the abstract when I was finishing up the E. coli. So after that, I was doing the Salmonella. So that's why I didn't mention the Salmonella graph into the presentation. But I got the most log reduction for Salmonella other than the E. coli.
Salmonella. And this plasma is under the the atmosphere pressure or the low pressure?
Yes, atmosphere.
Okay, and then the after you treated this beef, and then did you check any kind of the sensory change, kind of the texture or flavor, or actually like the lipid oxidation, rancidity?
I checked the temperature, surface color, and the moisture content of the beef. So there was not any significant differences before the treatment and the after the treatment.
All right, thank you.
Thank you.
Hey, Nadi, Mary. So non-thermal processing technologies have been around for a while and are being evaluated in the US and around the globe. Is there anywhere throughout the world where it is commercially being used on products?
The non-thermal techniques commercially used for medical technology, not for the food processing yet, but still on the researches. I hope in near future the commercial products will come.
Thank you.
Thanks, Nadi. Any questions for Nadi from the audience?
Nadi, I think you mentioned it, but I just wanted to confirm it. What type of plasma system was it? Because there are so many different plasma systems. And
yeah, it was the plasma leap system.
So is it more like having a jet plasma, or it's more like having...
No, it's having the pin reactor, There is many pins under the plasma chamber.
Oh, okay. And the conditions that you used is it something that you optimized to do it, or how did you decide on the conditions of the plasma treatment?
I checked with different parameters before, and then I selected some optimized things, and after that.
Okay.
Maddie, I have one question for you in the chat and I can read it out for you. Does the treatment penetrate the surface? That is, can it be used to treat ground beef?
Can you please say it again?
Yes. So the question is, can cold plasma treatment actually penetrate into the surface? For example, if you're using ground beef, can it be used to treat?
Yeah, Yes, we can do that.
Okay. Any more questions for Nadi at all? Oh, I see one more in the chat right here. Nadi, so would you explore different gases and see if you might get more log reduction?
Yeah, we can do that. But honestly, I did not study about it.
Okay, no worries. Thank you so much, Nadi, for tackling all the questions. And while the judges are grading, we can go back to our trivia and let's see who takes the trivia cup home tonight. What do you think? Which of this is not a true cereal grain? Buckwheat. This was tricky, to be honest. Ooh, we see some changes here. Think. What do you think? Yeah, it was actually a bunch of grad students and Dr. Gideon Livingstone. Good job. Cheese Master back on top. Okay, so assuming that the judges are done with grading, we can then move on to our last speaker for today. So our last speaker for today is Kylene Glews. She's a doctoral student studying nutrition at the University of Minnesota. Her talk for today is titled Bringing Back Our Old Friends by Eating Fermented Vegetables. Welcome, Kylene, and the floor is all yours. You may now share your screen. We can see your screen now, Kylene, so whenever you're good to start.
Okay, you can see everything? Sorry.
It is still, okay, yeah, now it is in the correct mode. So we can
Okay, perfect. Okay. Well, thank you so much for the introduction. We'll go ahead and get started. So today I want to talk to you about why you might be having increased feelings of loneliness. And no, I'm not talking about the loneliness we all feel due to the pandemic. What I'm talking about are some long lost friends in the form of microorganisms that scientists believe are missing from our diet and from our gut. These microorganisms, like fungi, bacteria, viruses, and others that live in the human body, are known as the microbiome and are essential to human health. However, due to a highly industrialized food system, as well as our busy lifestyles, packaged meals have become a major part of the daily diet. Furthermore, microbes that naturally are found on fruits and vegetables are often washed and packaged in order to prevent pathogenic outbreaks as they travel. For example, the strawberries we get in from Mexico to us here in the Midwest. Scientists believe this loss in beneficial microbes has contributed to chronic inflammation. In fact, the old friends hypothesis states that exposure to non-harmful microbes in our food and our environment is an important source of stimuli to help balance our immune system. So my research is focused on how we might be able to get our old friends back by researching fermented foods, specifically lacto-fermented vegetables. We hypothesize that the gut microbiome of people who regularly consume lacto-fermented vegetables may harbor more beneficial bacteria in their gut. So we enrolled 23 healthy human participants who include ferments like sauerkrauts and kimchis as a part of their diet at least 3 times a week. And we compare their gut microbiome to 24 individuals that say they never consume them. The results demonstrate some interesting differences between the fermented consumers versus the non-consumers. So for example, we found that Leuconostoc mesenteroides, which is a species of lactic acid bacteria found in sauerkraut, to be a discriminating bacterial signature in the gut microbiome of people that consume lacto-fermented vegetables. Certain strains of Leuconostoc mesenteroides have been shown to exert antimicrobial properties. We also observed the fecal metabolome, which are the small molecular compounds that are the byproducts of both microbial and human metabolism of food, to be highly diverse in the consumers compared to the non-consumers. This may indicate that fermented vegetables provide both live organisms and metabolites that are highly active with communities already established in your gut. Together, they may produce an even higher number of beneficial compounds, including short-chain fatty acids and other metabolites that have the ability to help maintain a healthy gastrointestinal tract and, of course, a healthier immune response. So implications from this study give us tremendous ideas for future research. If the regular consumption of lacto-fermented vegetables are able to provide more beneficial microorganisms and metabolites, should there be a daily recommendation as a part of the dietary requirements? What about our food system? Is it possible to ensure a food system that is safe, but also gives us exposure to these beneficial microbes? Certainly much more research is necessary. In the meantime, taking note of the microbial contents of your food in your daily diet may give you insight into what is working in the relationships with your little microbial friends. Thank you.
Thank you, Kylene. That was a fantastic talk. You may now stop sharing your screen and we can open it up for questions for Kylene, beginning with the judges.
Hey, Kylene, Mary, just really enjoyed your presentation there. I'm trying to think, what am I going to put on my hot dog tonight? Is it like sauerkraut? Now, do I get the sauerkraut in the jar, or do I get the sauerkraut like that's refrigerated, like individual pouches, or, you know, what do we know about all these, even just different types of sauerkraut, or sauerkraut versus kimchi, or should I throw some pickles on it and olives? And
those are great questions, Mary. And actually, I mean, so that was part of it. I mean, we have, you know, in order for us to have the shelf stable foods, we have really pasteurized everything. But I think that's one of the problems that we're running into. So definitely the refrigerated sauerkraut, if you can find the raw refrigerated, which there definitely are some now. And then that's the other thing that we're looking at specifically is how do things like sauerkraut from kimchis to just regular like fermented carrots and peppers and, you know, lots of different kind of fermented food. What specific strains in those would be beneficial or considered that sort of probiotic bacteria? So,
but definitely try and buy the refrigerated ones.
Definitely try and buy the refrigerated if you want your microbial friends back.
Okay.
So, you know, Kylene, it is a great presentation. So originally, I just came from the South Korea. I'm a very kimchi person. So I'm very familiar with the kimchi fermentation procedure. So usually the, you know, fermentation food, example of the kimchi. So the very early stage of the fermentation, the majority, the lactic acid bacteria is different. And then the later or the kind of the 1 week after or 2 weeks after, so majority microorganism is changed to yeast, and then their fermentation mechanism is a totally different pattern. So I just wondering this consumption, the fermented vegetable consumption for the intestine health, there is no any difference between the fermented the period. If someone is just eating the very early fermented food, or someone is eating the very late fermented food, that is no different.
Yeah, I think the research, we're still trying to research some of that, you know, Dr. Lee. I think that definitely as time goes on, there are going to be those microorganisms, certain microorganisms that take over that may not have as much beneficial probiotic qualities. But I think right now that's what we're all sort of trying to figure out. I mean, there was right now there's even a call of, you know, really like we can't necessarily call our fermented foods probiotic foods and that certain definition, because we don't actually know what strains are really in those, and especially during the different processes. Of course, we know that there'll be a lot of microbial activity happening in, you know, like you said, the first week of fermentation process. some studies say that it really kind of stops around day 5 or day 7. But those are great questions, and definitely that was the other thing. What kinds of sauerkrauts were these people eating, especially to see some of those specific probiotic bacteria that we find in the fermented foods. So
yeah, I really appreciate you just mentioned about the kimchi.
I love kimchi so much.
Thank you, judges, and thank you, Kylene. I have 2 more questions for you in the chat that I can relay right now. The first question is, what was the criteria for healthy participants, or what were the criteria for healthy participants?
Yeah, so that's a really great question. We had a bunch of criteria in that would eliminate them. Like, for example, if they had any gastrointestinal issues, if they were taking any sort of NSAIDs. I mean, there was a criteria really that people really weren't taking any medications. We also asked that people weren't really eating other fermented foods because we really wanted to focus on lactic acid fermentation of vegetables. So it was important to have the criteria that you really did include lacto-ferments in your diet at least 3 times a week, but you weren't really eating a lot of other fermented foods. Hopefully that answers the question. I mean, there was a whole list of of criteria that we made sure, people, I mean, of course, this idea of what healthy participants were, but we really wanted to make sure people weren't taking any other medications.
Yeah, that makes sense. The second question was, can the intestinal microbiome be changed by consuming lacto-fermented dairy products or from fermented vegetables?
Oh, I'm sorry, what was the end of the question?
So can it be changed by consuming lacto-fermented dairy products, or is the change possible only by consuming fermented vegetables?
Yeah, so great question. I think we have actually a lot more research showing that fermented dairy products definitely impact the gastrointestinal tract or have those sort of probiotic potential that we see with lacto-fermented vegetables as well. In terms of, changing the microbiome, my thought is that it probably isn't just one, dairy, and it's not just vegetables. It might be all of the products, like our sourdough breads, and really thinking about how we can get microbes, like I said, from even the environment back into our foods. So maybe not just dairy or just vegetables, but maybe a mix of a lot of different things in terms of the microbial content of getting it back in our diet.
Looks like moderation is the answer, and balance.
Yeah, moderation, balance, and lots of diversity, right?
Thanks, Kylene, for the great round of Q&A. And that concludes all the talks for today. Congratulations to all the speakers for all the hard work and effort that you have put. We will be announcing the winners shortly, but before that, we also need to find out who takes the trivia cup home. So let's go on and finish the trivia round for today. Wow, everyone got it right. A lot of cheese lovers in the crowd. Cheese Master, back on rice. This is our last question. Caramelization. Okay, so let's see. At the 3rd place, we have Jacobo. Congratulations. 2nd place, Lauren. And the 1st place goes to Cheese Master. Congratulations to all our 3 trivia winners. We will be sending you gift cards. So please put in your email IDs in the chat. We would also like to inform you that we will be sponsoring your nomination fees. So you can also get in touch with us if you're not a member of Phi Tau Sigma UMN chapter yet. We're happy to walk you through it, and we'll also sponsor your nomination fee. So congratulations to all the 3 trivia winners. And now we have a fun fact question for all the trivia winners. So Cheese Master, who are you? Congratulations.
It is me.
Sarah, of course, chewing down on cheese. I kind of guessed it. Congratulations, Sarah. So we have one fun fact question for you. Which was your favorite food science class and why?
I would say probably the sensory class. So the sensory evaluation of foods. Just because Zeta's in charge, Professor Vickers is in charge of that. And I like to think she's kind of my spirit animal.
Wow. Thank you for sharing that with us and congratulations on your win. The second place winner, Lauren, we also have a fun fact question for you. Which is your favorite cuisine and do you mind sharing why?
Probably like Mexican food. That's kind of like what I grew up eating as like comfort food, home food. So.
Okay, maybe that is why you started with salsa chips.
Yeah, yeah, exactly. That's my chips and salsa love.
Yeah, we can see your connection now. Congratulations on winning the trivia. Please share your email ID with us and we have some gift cards for you. And our third place winner, Jacobo.
Hi.
Hi, congratulations. We also have a fun fact question for you. Which is your favorite food item to cook or bake?
That's a good question. I would probably have to say pasta, just because I can do so many things with it. And every time I make it, I'm just like so thankful that I made that decision. So yeah, I'd say pasta.
Thank you. Yeah, pasta is very versatile. You can like eat it any time of the day, any day. Congratulations, Jacob. I hope you've shared your email ID in the chat again. And all the trivia winners, we will be in touch with you to get your gift cards and also tell you more about the sponsorship fees. So with that, I hand it over to Sonali. And fingers crossed for the most awaited moment.
All right, we are back and thank you for that wonderful trivia and thank you for participating in it and congratulations to all the 3 winners. We'll keep in touch with you. So the final awaited moment of announcing the winners. Are you guys ready to know who it is? And please do share in your comments or give some reactions to them so that all the participants are like happy with their presentations. And congratulations to all the finalists. We had a really close by scores for everyone from the judges. So you all really did a great job. So here we go. And are we ready now? And the third place for Speedy Science 2021 goes to Kylie. We have the second place for the Speedy Science 2021 goes to Kavya And the most awaited winner of the Speedy Science 2021 goes to, I don't have a drum roll, but yeah, drum roll. Rebecca. So once again, congratulations to all the finalists who has been part of this competition. And we have some cash awards prizes that will be sent to you, as well as certificates will be sent for all the 7 finalists for being top 7 of our competition. And the first place will be receiving $150 and the second place will be receiving $100. and the third place will be receiving a $50. And all the 7 finalists will also have their nomination fees waived for Phi Tau Sigma. So congratulations, everyone. And we will keep in touch with you guys to get your checks, as well as we would need your addresses as well. So our treasurer will be in contact with you. And once again, congratulations to all the winners and the finalists as well. And once again, I would like to thank everyone for their support for making this happen. And I would really like to thank AgriPure, as well as Dan and Erica Kennedy, for being our sponsors for this competition, and awesome judges for taking their time and quickly grading everyone virtually. So thank you so much for the judges and also the participants. And we hope you enjoyed the trivia round as well. And finalists, everyone's presentation was amazing, and you guys were amazing today. And finally, I would like to thank the Phi Tau Sigma officer team as well. We all did a good job. And yeah, see you everyone back at Speedy Science 2022. If you have any questions, feel free to email us. If you'd like to join the Phi Tau Sigma Society, please email us and we'll guide you through the process. And we hope you enjoyed Speedy Science 2021.